Okay, welcome back everyone to the second session. It has a very broad title, From Structure to Function. We have 4 short talks, and the first is from William Podlaski.
Okay. Hi everyone. So today I'm going to tell you about computing with spikes. And just to start, I think it's safe to say that many of us in the Cosyne community have gotten used to thinking about dynamics and computation in more abstract rate networks, but I'm going to try to convince you that there's still some conceptual differences in how to map that to spikes, and I'll try to give you a new perspective on how we might do that. So let's start with the typical picture of computation through population dynamics. Here is a trajectory in neural state space, and we can imagine that this activity lies on a low-dimensional manifold, as is often observed in data, so that we can understand the trajectory and dynamics in a lower dimensional state space that I'll call the latent space, Y. So here we have the dynamics in Y1 and Y2. So we know that we can model these dynamics using an RNN with connectivity W, and we can operationalize these low-dimensional dynamics by using a low-rank network. So here I'm going to use the notation W equals ED. So we have these 2 rectangular matrices that are the encoder and decoder of the recurrent weights. So essentially we can understand the recurrent weights as passing the activity through a bottleneck. So first the neural activity is decoded into the latent space, and then it's encoded back into the inputs of each neuron. So what I want to focus on here is how do the underlying rate and spiking dynamics fit into this population level picture? So let's start with rate neurons, which we understand pretty well. So let's imagine that we have a network of ReLU units. So the encoding weights of this recurrent connectivity basically tell us where each of these units will be active. So the encoding weights will kind of orient the ReLU activation in the space, and then in one half of the space this unit will not be active, in the other half it will have a linear activation, shown here in 3D. Then we can put another neuron in this space with different parameters, and so on. Then we go to the decoding weights. So the decoding weights basically tell us what are the dynamics in this space. So if there's any area of the space where there's no activation, we basically just have subthreshold leaking dynamics. But then the interesting thing happens where the neurons are active, and basically each neuron's decoding weights contributes some type of vector field to the population dynamics. So there's one neuron, then we can add another one, and in total we basically get a flow field where we can visualize trajectories as flowing through these different areas of the space. And from this we know that, from a lot of nice previous work, that these types of networks can generate arbitrary dynamics. So, okay, let's move on to spikes. So how do spikes change this picture? So here's a depiction of a spiking neuron. Its voltage is integrating input, and then it's occasionally emitting a spike. So it turns out that one way of representing an idealized form of a spike is by thinking of its nonlinearity as this so-called indicator function. So essentially this neuron, when its voltage is below threshold, it's emitting no output, and then briefly at times its voltage will go above threshold, where it will emit plus infinity output instantaneously, and then it will return back down. So it turns out that if we try to put that into this picture in latent space, each neuron's nonlinearity basically contributes this sort of infinitely high wall in the space. And we know that the voltages are only going to be momentarily above threshold, so this area of the space essentially becomes this forbidden zone. This indicates that one or more of the voltages are sitting above threshold, so we know that the activity isn't really going to be lingering in that area of the space. So what do we get from this picture? Well, we see that this idealistic spiking nonlinearity poses kind of a puzzle for computation, because the very area that the ReLU units were using to implement arbitrary dynamics is the area that we actually can't use in a spiking network. We are only left with this boring subthreshold area. So of course, there's been a lot of great work in dealing with this, and I would say the most common approach is a smoothing approach, where you basically just take this difficult nonlinearity, smooth it into rate dynamics, and then work with that, right? But I would argue that that's kind of, throwing the baby out with the bathwater a bit, right? Because then you're removing the actual spiking dynamics from the picture and assuming that the spiking neurons are following rate dynamics. So today I'm going to tell you a little bit about a different approach that I'll call a spike geometry approach. So to start with, let's take this two-neuron picture and actually just simulate the dynamics to see what we get. So when we do that, we see that we do have stable dynamics in this space. Here's the voltage plotted of each neuron over time, and then the latent variables. And we can observe a couple things from this picture. First of all, we get stable dynamics at the intersection of these two areas of the space, and that's essentially because I designed this network to actually have stable dynamics, and the key thing is that these decoding vectors have to point into the subthreshold area. So we basically have dynamics that moves into this forbidden zone, but then is pushed out by each spike. And this turns out to correspond to a mutual inhibitory network. The second thing we can notice is that in this two-dimensional latent space, we actually only have a one-dimensional nonlinear manifold that we can actually work with and build computations on. So we've somehow constrained the space in the area of the space where that we're actually able to build dynamics. Okay, so it turns out that this intuition applies then for scaled up networks. So let's see what happens when we add more neurons and then more dimensions to this picture. So it turns out that if we constrain the recurrent connectivity to be inhibitory, we can actually build a network with the same type of population level threshold between subthreshold and superthreshold areas, like so, where I'm just highlighting where each neuron is contributing in this area of the threshold. So there's each neuron is coding for a little area of this manifold. Then for the dynamics, each neuron's decoder should point in the direction of the subthreshold activity, but we have a little bit of flexibility in how we want to orient them. So it turns out using that, we can basically implement dynamics on this manifold. So as one example, we can put a bistability in this system by putting two fixed points here with an unstable one in the middle. And when we simulate this network, we essentially get dynamics that follow this bistability. Okay, and it turns out that we can kind of extend that to any arbitrary one-dimensional dynamics in this model. Okay, so what about when we add extra dimensions? So if we consider one more dimension, we get a picture that looks like this. So now we have a three-dimensional latent space, and we have a two-dimensional surface manifold in this space. Every neuron is essentially one of these little squares. I'm only showing where this neuron is contributing more than the other neurons to this manifold. And then we can implement two-dimensional dynamics. Here is shown for a stable limit cycle, and we can simply simulate this network. And here the decoders have basically been fit to approximate these dynamics. Okay, so one additional thing we can do here that already allows us to relate this to biology is think about what it means that every neuron is only coding for a local area of this space. So it turns out that when neurons are removed from this network or inhibited, the neighboring neurons can fill in and do the same job. So what I'm going to do now is kill 30% of the neurons in the network, and essentially what happens is some of these little squares will be removed, and you see that the other ones kind of fill in the space. And then we can simply simulate the same thing. So here the network hasn't been retrained, it's just removing 30% of the neurons and simulating the same thing. We see that we get nearly the same dynamics out. And I would say if you did this in a ReLU network or a rate network, it would be very difficult to remove 30% of the neurons and get the same dynamics out. Okay. So then in the last bit of the talk, I just want to start talking about excitatory neurons. How do they fit into this picture, as promised in the title? So it turns out that we can think about excitation in this picture as well. So going back to this one neuron picture in 2D, an excitatory neuron is essentially a neuron that has its decoding vector pointing in this superthreshold area, which essentially creates a positive feedback loop. So when the dynamics move into this, cross this boundary and the neuron's voltage hits threshold, it's just going to keep firing spikes along this axis indefinitely because it doesn't have any way of stabilizing itself. So what we can do is that we can then put an inhibitory neuron in this space that then creates an axis of stability, such that when we combine them, we basically get stable dynamics at this intersection where we have an excitatory spike closely followed by an inhibitory one. So it turns out that this motif can then be used to build dynamics by adding more dimensions to this picture. So here's a three-dimensional space now formed of two excitatory dimensions that now have a global inhibitory dimension that stabilizes their activity. And when we simulate this network, we can basically implement dynamics along this one-dimensional manifold. And here, as you can see, the cross-section of the dynamics are shown here, and we basically have this type of inhibition-stabilized dynamics along 2 of the dimensions, and the 3rd excitatory dimension is free to implement dynamics. Okay, so with that, I'll summarize. So what I've shown you here today is a spike-based view of computation through population dynamics, and it's one along one of several works that have used a low-rank approach to model dynamics. Some of them in rate models, some of them in spiking models. I would say that ours is unique in that we use this spike-by-spike approach, meaning that we can account for the role of every individual spike. We don't need to use large networks or any averaging, and so we think that offers some advantages and also more relation to the brain. And from this spike-based operating regime, we get some interesting relationships to biology that I'll just highlight a couple here. One of them is that we can sort of think of neurons as boundaries. So I think that this is somewhat related to work in rate networks and thinking about neurons with steep non-saturating nonlinearities. I've already mentioned that we get robustness from this picture. We also get, as I mentioned, directions in latent space that are constrained. So we actually can't use all of the dimensions of latent space to implement dynamics, but some of them are constrained to just control the activity. And overall, we think that we're approaching some more computational explanation of what Dale's law is for, because it sort of emerges naturally from this framework. So with that, I'll finish. If you're interested, most of this work will be put in a preprint form soon, but we already have work published last year on the rank 1 case that you can check out, as well as some work on scaling this up to associative memory networks. Thanks very much.
Questions?
Thanks for the great talk. I was curious, is there any relationship between the dimensionality of the input and the number of neurons in your network?
Yeah, so
basically,
I didn't talk much about the input here. So input basically offers another layer of complexity, so we can have any number of input variables that then the neurons can have various tuning to. So if you change the input, you basically can move the neurons around in this space, and that gives you some extra flexibility. But it's a kind of an independent parameter related to the number of neurons.
Thanks.
Hey, your latent variables are expressed in rates, essentially. Is that right? So what happens when you express those in spikes?
Interesting question. Yeah, I think we're still at the point of trying to build continuous dynamics into these networks, because essentially that's our standard. So if we want to actually convince people that we are implementing dynamics, those will be spikes approximating smooth continuous dynamics. But I think it's a very interesting question to think of if we actually have discontinuous spiking kind of dynamics in latent space, and that's actually the goal of the system, that maybe we can do more interesting things. So thanks for the question.
We have time, so I'll ask a question.
Okay.
So you made a dichotomy between the smoothing approach and the spike-by-spike analysis. Could you say a little more about what you would miss if you use the smoothing approach to understand the problems that you described today?
Yeah, so I guess maybe going back to this beginning slide. So I think if you try to build a spiking network using a smoothing approach, you can essentially arrange the neurons in the latent space using any arbitrary geometry that you might want, which means that you may have some areas of the dynamics where you would get quite a high spiking because the neurons are essentially being driven strongly above threshold. So I think the difference with this approach is that it allows you to constrain the geometry in such a way that this limits the spiking. So it essentially changes the geometry a bit. And in principle, you could arrange the neurons in a rate network or in using a spiking network with more of a smoothing approach to also do that. It's just that you wouldn't maybe think that you would have to constrain it in that way without taking this view.
All right. Well, if there's nothing else, thanks again. Our next talk is from Chengcheng Huang.
Sorry about that. I want to thank the organizers for the opportunity to share our work here. We know that our perception can be strongly modulated by the internal states of our brain. For example, what we see in the image strongly depends on where we pay attention to, our expectations, motivation, and etc. The internal states of the brain also strongly modulates neural activity. For example, in this work from the mouse visual cortex, it is showing that when the animal was running and in a more alert state, the average firing rates of neurons were higher and the average correlations between neurons were lower. So we are interested in studying how the internal states modulate neural responses. It has been hypothesized that the inhibitory interneurons play a critical role at regulating state-dependent neural activity. Traditionally, neural network models only consider homogeneous populations of excitatory and inhibitory neurons. However, we now know that there's a huge diversity in the interneuron cell types. So here we consider three major interneuron cell types, and they are PV, SOM, and VIP neurons. These interneuron subtypes have distinct connectivity patterns, and they are targeted differentially by feedback connections and the neuromodulators. Therefore, they are well suited at regulating network activity states. So the goal of this work is to study how modulatory inputs to different cell populations modulate firing rates and network synchrony. So we studied this in a spiking neuron network model. So all the details can be found in our recent preprint on bioRxiv. Our model consists of 4 neural populations, the excitatory population and the 3 interneuron populations. The recurrent connectivities are based on anatomic data from layer 2-3 of mouse visual cortex. In particular, we assume that there's no connection between SOM and other SOM neurons, and no connection between VIP and other VIP neurons. The feed-forward inputs are modeled as Poisson spike trains, and they only target E and PV neurons based on findings from the mouse visual cortex. The neurons in our model are distributed randomly on a 2-dimensional plane, and the connection probability between neurons decay with distance. In particular, we assume that the connections to and from the SOM neurons have a larger spatial footprint compared to other connections. The larger spatial footprint of the SOM neurons are believed to be important for surround suppression in visual cortex. And lastly, we apply static current to one cell population at a time and measure changes in neural population activity. The static current models for top-down modulation. As we apply varied inputs to different cell populations, we consistently observe 3 representative network states. So first, in the subcircuit asynchronous states, the SOM neurons are silent, which decouples the VIP neurons from the E and the PV neurons. Therefore, the network is reduced to E and PV subcircuits, and their dynamics are asynchronous. In the weak synchronous state, we can see coordinated silence period in E and PV neurons, which follow rapid firing of the SOM and the VIP neurons. In the strong synchronous states, the network exhibits strong oscillations. In each oscillation cycle, E and PV neurons fire first, and followed by SOM and VIP neurons. So we first look at the case where we apply input to the PV neurons. We found that when PV neurons are inhibited, the network is in a strong synchrony state. And as we increase the input to the PV neurons, the network synchrony level measured by the maximal coherence between neurons gradually decreases. The firing rates of the E and the PV neurons show non-monotonic changes as we vary the input to PV neurons. In particular, during the weak synchrony states, the excitatory neurons' firing rate increases as we increase the drive to the PV neurons, which is because of the large reduction in inhibition from the SOM neurons. So we can summarize the patterns of modulations in this phase plan of the firing rates and the maximal coherence of the excitatory neurons. As we apply input to a cell population, it moves the network states along a certain path in this phase plan. For example, as I just showed you, input to the PV neurons decreases the network synchrony and results in non-monotonic changes in the firing rates of the E neurons. Interestingly, input to the excitatory neurons moves the network along the same path, but with opposite direction. Increase the input to the excitatory neurons increases the synchrony level. Similarly, input to the SOM neurons also increases network synchrony, but it monotonically decreases the firing rates of the excitatory neurons. Lastly, input to the VIP neurons moves the network along the same path as input to the SOM neurons, but with opposite direction, and it never leads the network into the strong synchrony states, which can be an advantage of applying input to the VIP neurons. So we can see that in this network of 3 interneuron subtypes, the rate and the synchrony of the excitatory neurons can change in opposite directions. This is in contrast with the modulation patterns we see in the EPV subcircuits, where the rates and the synchrony of the excitatory neurons are tethered together to always change in the same direction as we apply input to E or PV neurons. We found that the SOM neurons' activity is most correlated with network synchrony. When we plot the maximal coherence of the excitatory neurons against the average firing rates of the SOM neurons, all the modulation paths across the 4 input cases collapse together. We see a consistent and a monotonic relationship between the synchrony level and the average firing rates of the SOM neurons. In contrast, we don't see such a tight relationship in other cell types. So this suggests that the SOM neurons are critical, the main driver for network synchrony. Our model is consistent with experimental results from the mouse visual cortex using photoinactivation. In this experiment from the Adesnik lab, when they inactivated the PV neurons, they observed an increase in the power spectrum of the local field potentials across frequency. Our model also predicts an increase in the coherence level. In contrast, when they inactivated the SOM neurons, they observed a large reduction in the power spectrum, in particularly around the low gamma frequency range. Our model also predicts a reduction in the coherence. In a later work from the same group, they also observed a tight relationship between the SOM activity and the overall gamma power across stimulus conditions. So here, each dot is a visual stimulus condition of different contrast and sizes. So we can see that the average activity of the SOM neurons is highly predictive of the overall oscillatory power over the low gamma frequency range. Next, we investigate the impact of SOM connections on modulation patterns. First, we found that strong inhibition from SOM to PV neurons leads to abrupt transitions from asynchrony to strong synchrony. We can see large jumps in the coherence level with small changes in input values. Moreover, we also see that the rates and the synchrony tends to vary in the same directions in these networks, which is qualitatively different from the modulation patterns in networks where some neurons inhibit E more than PV neurons. Second, we found that the E to some connections are critical for some induced network synchrony. When we narrow the spatial footprint of E to some connections, we can see that the maximal coherence achieved in this network with modulation is much lower than that in the default network with broad sum connections. In contrast, when we narrow other sum connections, we see no changes in the coherence level. So to summarize, we found that the firing rates of some neurons are most correlated with network synchrony. Inhibition from some to PV neurons must be limited to allow gradual transitions from asynchrony to synchrony. And E to some connections are critical for some induced network synchrony. Lastly, I want to thank my group members, collaborators, and the funding sources. This work was led by my former postdoc, Madeline Edwards, and was in collaboration with Jonathan Rubin from the University of Pittsburgh. And thank you for your attention. I'm happy to take questions.
Questions?
Hi, thank you for the talk. Do you see any spatially structured activity patterns like traveling waves in your network?
I didn't include the movies. So because of the spatial structure in the network, we see kind of irregular propagating waves. So we don't have very periodic traveling waves in the weak synchrony regime. And then in the strong synchrony, it's kind of just spread out very fast. Thanks.
So you highlighted one or two key parameters that determined whether you had this kind of discontinuous transition to synchrony. Are those the only parameters that matter, or are those somehow the parameters that kind of take you across the transition most readily?
For the connection weight?
Yeah.
Yeah, so far our analysis mostly focused on connections to or from the sum connections. We found is the sum inhibition, the relative inhibition to E versus PV neurons, that's very important. Basically changes the transition from like supercritical to subcritical transitions. And the strength of E to some connections are also important. We haven't exhausted the parameter analysis in this network.
And are the VIP neurons playing a role, or is it really SOM, PV, and excitatory there?
Yeah, so we found that the VIP neurons are not very critical in the state transitions, basically because their interactions are all mediated through the SOM neurons. So if the mutual inhibition between VIP and the SOM are very large, then we can see sharp transitions just because of this mutual inhibition, then the transition can be sharper. Thank you.
Anything else? All right.
Do you want to try this?
Next talk is from Kevin Sitt.
Great. Hi, everybody. I'm Kevin Sitt. I'm from the Adesnik lab at UC Berkeley. And I'm happy to share with you some work that I've been doing, kind of like extending all optical interrogation to try to give us kind of unprecedented control of neural activity to answer a lot of cool, interesting questions. But today I'm just going to share one of these stories with you. And that's on the stimulus quenching of neural variability. So first, let's get some background. So the brain shows a remarkable amount of variability. This manifests on the population level with a huge heterogeneity of neural responses, but it also manifests at the single neuron level, where a neuron shows variability in its response even to the exact same stimulus over many trials. So that's shown here with all the individual trial traces shown in orange, and then you can see there's a ton of variability. But one other thing you might notice is that when a stimulus comes on here at the same time, we get a reduction in variability across trials. We get a reduction in this intertrial variability, We can use the Fano factor, which is defined as the intertrial variance divided by the mean, to try to capture this. And when we look at the Fano factor, when the visual stimulus comes on, we can see a sharp decrease. This is recorded from neurons in area MT to this kind of plaid stimulus. But it's important to note that this isn't just for one type of stimulus or even one type of brain area. We actually see the stimulus-driven quenching of neural variability across many brain regions, different stimuli, and even different sensory modalities. So because of how widespread this is, we think that understanding some of the mechanisms that drive this phenomenon might provide really important insight to kind of a fundamental property of neurons and help us generally to better understand the brain. So what are some mechanisms that might explain this? So in really broad strokes, we could imagine that one possibility is that the reduction in this variance might be inherited by the neuron from its network. We can imagine that a stimulus presentation, the onset of a stimulus, drives more similar patterns of network activity across trials, which reduces the neuron's response variability. Indeed, there's been a lot of computational work that's suggesting plausible network models that result in this phenomenon. But are there other possibilities? We could also go to the complete opposite side and imagine a cell-intrinsic phenomenon, something about the cell itself that might drive it to kind of respond less variable when a stimulus comes on. In this case, it doesn't matter what's happening in the network, just something about the stimulus coming on will drive this reduction in variability. So the first question I want to get at is the stimulus quenching variability of this network or cell-intrinsic mechanism? So how can we get at this question? So we can first look at whether or not it's a network phenomenon just by measuring cells and looking at their variability and then the variability of the inputs that are going into that cell. So we can take mice, head fix them, and then have them view visual stimuli. Oh, I do want to note, this work was done by Chengcheng, who just gave a talk, Yulia and Hillel. So we head fix mice, we have them watch a visual stimulus. First, we can just measure spiking of a neuron, and we can see, as expected, when the stimulus comes on, we get a reduction in the variability. Great. In this case, a reduction of the Fano factor. So next, we can patch onto a neuron and measure its excitatory and inhibitory conductances, which are a proxy of the inputs that it's getting. Well, it actually is the inputs it's getting. If we expect that there's a quenching of variability in the network, we would expect a reduction in the variability in these excitatory and inhibitory conductances, but instead we see the opposite. When the stimulus comes on in both the excitatory and inhibitory conductances, we don't get a quenching of the variability, instead we get an increase. So the synaptic input variability is not quenched. I do also want to note, I don't have time to get into it now, but Chengcheng has also worked to show that this is not explained by changes in the covariance of the excitatory and inhibitory conductances. So because the variability increases on the stimulus presentation, these data strongly suggest that this kind of reduction in variance on the output is not due to some kind of changes in input, it's not inherited from the network. So because of that, we need to consider what cell-intrinsic mechanisms may be at play here. So we think of something really simple. We can imagine all neurons must abide by Ohm's law. So we can extend Ohm's law from kind of means to variances. Quick note, this is of course not a proper derivation, but I think it provides reasonable and valuable intuition. If you're interested, please keep an eye out for the preprint that's going to be coming out with a formal derivation, as well as a lot of computational and theoretical support for this idea. But I think the key point here is that we can see, or we can expect, that the variance of the membrane potential might decrease with increasing conductance G. So this leads us to a really key prediction and a hypothesis, that increasing conductance to a neuron, total conductance, doesn't matter if it's E or I, is sufficient. It's alone sufficient to quench its variability. And we really wanted to test this in a living mouse in vivo. So in order to test this hypothesis, we needed to be able to inject conductance into a neuron, but we also wanted to minimize network effects because we didn't want kind of these confounds of something going on in the network. To do that, we used holographic two-photon optogenetics to inject, or we can use two-photon optogenetics to inject excitatory conductances usually into a cell. I'm just giving you an example here. So what you'll see is you'll see flashes of activity, or flashes of light, which are activity, and we're essentially writing a pattern of activity into this mouse's brain live. So you can see we're drawing the letter A into the mouse's brain. Not sure what the mouse is thinking at this point, but we think this at least demonstrates the really high spatial temporal precision we have here. But there's kind of a little bit of a problem. If we take a neuron and we inject a bunch of excitatory conductance into it, we might drive the activity of this neuron into its saturating regime, which will artifactually reduce the variability across trials. On top of that, if we drive a neuron really, really strong, we might maybe cause some kind of small network effect from this neuron kind of going crazy. So how do we get around this? The trick is to using a balanced inhibitory conductance to bring this neuron back to where it was before. Essentially, what we're trying to do is inject a large amount of conductance, but because it's balanced, we get no change in the neuron's firing rate. So how do we do this? Luckily, recently, there was a new kind of opsin construct that was developed by the VROC lab that combines an excitatory channel and an inhibitory channel that are separated spectrally. What this allows us to do is have independent control of both excitatory and inhibitory conductance. And I do want to mention that these channels, the ions they let pass, mimic the natural channels found in the neurons. So this is awesome. We have this awesome protein that we can use, but what was really lacking was the means to perform actually two-photon holographic manipulation in multiple colors, in two independent colors, so that we could control these two at our will. So that's what we developed. We developed the first multicolor holographic optogenetic microscope. This kind of schematics for aficionados, I'm happy to chat with people in detail about it later, but the key takeaways is that we get independent multicolor holographic manipulation. So we can target the same cells, we can target different cells, the patterns are completely arbitrary, we can change the power, doesn't matter. We get flexible multi-neuron ensemble targeting, and of course we get the same high spatial temporal precision as with single color stuff. Okay, so both colors are technically in the infrared kind of spectrum, but I'm gonna refer to them as red and blue, so that's for simplicity. We can imagine the red one opens the excitatory channel, and the blue one opens the inhibitory channel. So more red light, we get more excitation, more blue light, we get more inhibition. Let's see the scope in action. So I'm taking a mouse here, it's expressing GCAMP, a calcium indicator in these neurons, that's green, and we express the Bible's construct that's tagged with a red fluorophore. So these kind of yellow cells have both. I'm gonna first shine red light on these three neurons. The color map here is, white is like no change, redder is increases in activity, and then blue is decreases in activity. We can see that when I shine red light, we get strong activation of these three neurons, and then next trial, when I shine blue light, I get strong inhibition of these three neurons. I can show you this another way, where we can start off here. This is just the cell's response to a weak visual stimulus. We use this so we had something to take away with the suppression. I can add increasing amounts of red laser power, which will increasingly drive more and more activity of this neuron by increasing its excitatory conductance. On the other side, I can also add more and more blue light to decrease its activity. So what I just showed you here is really this kind of the two arms of this potential matrix of all the powers we can try to use, and this really gets to where we can get that balanced kind of conductance. If I sweep across and try all these different combinations, we can see that there exists a combination, this kind of diagonal, in which there's no change in the neuron's activity, but we know we're increasing the conductance of this neuron. That is, although the activity here and maybe the activity here in terms of calcium or spikes are very similar, here the neuron has way more conductance, it's much leakier, it's in a completely different state than in this square here. So with this development and proving it that we can work, we can finally set about asking that question that we had, whether or not the increasing conductance alone is sufficient for driving the stimulus quenching and variability. Okay, let's get to that experiment then. So we take a mouse, have a view of visual stimulus, we're recording from neurons while we're using this fancy system to manipulate the neurons. First, let's just make sure everything's working as expected. Increasing contrast drives higher response, great. On the other side, increasing contrast reduces the Fano factor, also awesome. And now we're gonna just take one of these. So we're just gonna start at the same visual stimulus. We're gonna show it over and over again. We're gonna try adding our manipulation. So what I first wanna show you is this kind of like 2D histogram where when we show the visual stimulus, we get this mean activity in kind of white, but around it you can see there's a lot of variability of the responses. This blue bar is when the visual stimulus was on. So great, we get these responses. Then I'm going to pair that visual stimulus with our balanced optogenetic manipulation. So the first thing I want you to see is that there's no significant change in the mean activity here, but if you look at kind of like the spread of the 2D histogram, we've kind of shrunk it a little bit. We've reduced the intertrial variability without changing the firing rate just by hitting it with this balanced optogenetic manipulation. We can, of course, continually increase the total conductance by increasing our powers. If you remember that matrix, I showed you multiple squares which had the same no net change in firing rate. And if we do that, we can see a nice relationship between the amount of total conductance we're pumping into the cell and its Fano factor decreases. And I also want to mention that kind of like the scales we're looking at here are very similar to what you get in the visual conditions. So we really do think that these mechanisms are at play in both of these conditions. Okay. So in summary, I showed you that a balanced increase of total conductance of a neuron is sufficient. Without worrying about what the network is doing, it's sufficient to quench its intertrial variability. But the other thing I really want to talk about or highlight here is this kind of new tool of this multicolor holographic optogenetics, which I really think opens up a lot of interesting new experiments. You can imagine kind of things like if we know like an ensemble of neurons might be important for like some kind of behavior, we can do sufficiency and necessity experiments in the same animal. We could do kind of bidirectional coding in sensory cortices. We know many stimuli drive not just increases in activity, but decreases in non-overlapping patterns of neurons. So we can try to recreate that activity more faithfully. And of course, we can look at really interesting excitatory or inhibitory balances in vivo. So with that, I'd like to thank my lab, our collaborators, funding, and then my contacts up there. Feel free to email if you have interest in collaboration, have any cool ideas. We built this thing. We want to use it. And thank you for your time.
Start over there.
Thanks for the talk and the work. You started with showing those now classic results from, for example, Mark Churchland showing the reduction in Fano factor and variability with the stimulus onset. They also showed that it is mainly or entirely the shared variability across neurons in the network that goes down rather than private variability. Does this model, which ultimately is due to the reduction in the integration time, right, like the membrane time constant of the neurons, does it account for that? The reduction in shared variability being the main?
So I will say I didn't do any of the computational work, so I don't want to make a wrong answer, but my expectation would be that with this model, the increase in activity in any of those neurons would decrease the variability, or sorry, the increase in conductance to any of those neurons would decrease the variability. I'm not sure how to disentangle it being private versus shared, but I would imagine you would see similar effects just because each cell would experience that reduction due to the increase in conductance.
Thanks.
Yes, hi, very nice tool. I'm curious about the motivating experimental observation that you see a drop in Fano factor and an increase in the conductances. Because when I looked at it, it looked like the drop in the Fano factor preceded the increase in the variability of the conductances. So it's a bit hard to kind of rectify that with a causal role in the increase in the variance of the conductances to the drop in the Fano factor of the neuron. Could you just comment on that, please? Yeah,
so I think you're referring to the spiking data where it showed the Fano factor dropping when the stimulus was coming on. I think that's actually largely just a smoothing artifact. When calculating the Fano factor across time, we calculate in these binned windows. And because of that, kind of like right at the onset of stimulus, you get maybe a little bit of a reduction before the actual stimulus comes on.
Fair enough, but it seems like one could resolve that through like a careful numerical analysis, and it may be worth doing that.
Absolutely.
Thank you.
One more question. I think you were up first.
You mentioned in the beginning that the synaptic excitatory and synaptic inhibitory variability increased. Can you comment on the direction in which both of these excitatory and inhibitory synaptic inputs increase? Because if they are in the same direction, then it could be that they cancel each other out, and the net effect is that the variability decrease is still due to network effects.
Yeah, so in the full paper, there is work that shows that it's not due to this kind of like covariance or like a kind of like a correlated increase in the two. I don't have the figures with me here, but then I think or maybe Chengcheng will be happy to talk to you about it.
Thank you.
Thanks.
Thanks.
Our last talk is from Timothy Currier.
Okay, hello everyone. My name is Tim Currier. I'm a postdoc working with Tom Clandinin at Stanford University. So first off, thank you so much to the organizers for the opportunity to share my work and for putting together such a terrific meeting. Today, I'll tell you about my work exploring the capacity of connectome data to predict the functional properties of neurons. I'll tell you about some sparse recording experiments where I measured the visual selectivity of dozens of cell types in the fly optic lobe. And then I'll show you how well, or in some cases not well, the fly connectome does when making various kinds of prediction about my new data. So to get into it, the relationship between structure and function in the brain is a central question of neuroscience, and over the past 40 years, connectomes have emerged as powerful tools for understanding this relationship. One of the great promises of connectomics is the ability to predict function of neurons based on their morphology and connectivity. And many labs have done exactly this for some specific cell types with pretty good success. However, the overall functional information content of connectome data across cell types remains unclear. And with the recent publication of the full brain connectome for the fly, we can now try to address this big picture question. In particular, the fly visual system has a lot of advantages. Most importantly, complete connectome annotations. This highly stereotyped network is a series of stacked neuropils that sequentially process incoming visual information. And one feature of the fly visual system that I want to highlight is the big expansion in the diversity of anatomical cell types that occurs at the level of the medulla. The setup is actually very similar to the octopus that we heard about earlier from Chris. While earlier processing stages have only a handful of cell types, there are more than 100 cell types in the medulla, maybe even more than that, depending on who you ask. Because of this diversification, we decided to focus on this area when trying to assess the functional information content of connectome data. So if we want to ask how well connectomes can predict function, we'd like to have physiological recordings from many cell types to use as a reference data set. Really, the more cell types we can record, the better. And so I modified an existing genetic approach to express GCamp in an extremely sparse and random subset of neurons, as shown here. Because the expression is random, over many flies, I can sample a huge number of anatomically identified cell types. During these recordings, I presented flies with a noise stimulus like the one shown here. If you saw Greg's talk yesterday, this will look pretty familiar. Using the response to this noise, I could then calculate a fluorescence-weighted stimulus history to estimate the preferred stimulus of each neuron in the imaging volume. These preferred stimuli take the form of a spatiotemporal receptive field, or STRF. As the name implies, an STRF describes a spatial location and temporal dynamics of a stimulus that maximally excites each recorded cell. And so I performed these recordings over 54 flies, which yielded 510 unique neurons. Pretty good data set. These 510 cells come from 91 cell types found in the connectome. However, because this is a random labeling strategy, not all 91 cell types were well sampled. And so because of this, my subsequent analyses today are going to focus just on the 43 cell types that I recorded at least 3 times each. That said, of the 91 cell types in the full data set, at least 55 of these have not been previously recorded. So I think this data represents one of the most comprehensive accounts to date of early visual processing, and I'm really hoping it'll be a useful resource for folks in this room modeling visual computations. If anyone's interested in working with the data, please definitely come chat with me. Okay, so with this data in hand, we can finally start to ask some hard questions of the connectome. We began by sifting through the literature for examples of unvalidated connectome-based predictions of feature selectivity for certain cell types. Features that had been previously predicted include receptive field size, orientation selectivity, direction selectivity, and spectral selectivity. For the sake of time, I'm not going to linger on each case-by-case validation, but the overall message here is that the connectome does a pretty good job predicting most of these features. The one exception was spatial receptive field size, and we think in this case, subcellular compartmentalized processing is to blame. I'd love to dig more into this with anyone who's interested, but I encourage, if anyone wants to know more about these specific predictions, to check out our preprint. So again, these case-by-case validations I just mentioned were focused largely on single cell types, now let's hone in on our big picture question. I'm showing here a table of the connection strengths amongst all of the cell types in my data. Since we have recordings of all of these presynaptic cell types, you could imagine trying to predict the activity of each postsynaptic cell type. Perhaps the simplest way presynaptic inputs might be combined is by direct summation, with each input being weighted by the fraction of total input that it provides. By summing over weighted inputs, we get a predicted postsynaptic response that we can then compare to the actual measured response. And so here I'm going to quantify the accuracy of each prediction by calculating the correlation coefficient between the predicted and actual response time courses. And we're going to compare the full model to a reduced one that bases the prediction only on the single largest input. And so if neurons are actually doing weighted linear summation, we would expect the prediction quality to be higher in the full model. And while this was true for maybe a handful of cell types, we found that across cell types, the full model performs no better than the reduced model, somewhat surprisingly. However, we did notice that the correlations were already quite high for many cell types in the reduced model. This suggests that most cell types are each highly correlated with their largest respective inputs. This prompted us to explore the complementary reduced model, which bases a prediction on the weighted activity of all inputs except for the largest input. And here we do see a significant decay in the model's prediction quality, confirming that the brain's strongest inputs are playing some kind of privileged role. These results then suggest an alternative model to linear summation, such that strong inputs are heavily overweighted and weak inputs are essentially ignored. We found that this strong input dominant framework yields better functional predictions than weighted summation for more than 2/3 of the cell types in our data set, colored in yellow here. I should say that this is still an extremely simple model, and I'd really love to see how much better a more sophisticated model can do, and talking with all of you has been very insightful in that regard. Okay, but so direct prediction of postsynaptic activity from known inputs isn't a super realistic test case, as it assumes that you've got recordings from at least some inputs for your cell type of interest, and that's just not always going to be the case. And so let's see if we can give the connectome a harder task. Perhaps you see an interesting cell type in the connectome and notice that it shares some inputs with a different cell type that has been recorded. Under these conditions, can we assume that our interesting cell type will be functionally similar to the recorded cell type? This is essentially asking if similar connectivity is predictive of similar function. To address this question, we imagine cell types as living in a pair of high-dimensional spaces. The first space is a connectivity space where the dimensions are input connection weights. And the second space is a function space, which we define by performing principal components analysis on the STRFs for all recorded neurons. In these spaces, we can use the Euclidean distance between any pair of cell types as a measure of the relative connectomic or functional similarity between those two cell types. We can then ask if connectivity distance predicts functional distance in any meaningful way. So what I'm showing here is a clustered matrix of type-to-type connectivity distances. If we mirror this connectomic clustering onto a similar matrix of type-to-type physiology distances, we might hope to see some of the same structures emerging. However, the mirrored physiology distance matrix looks, frankly, like a hot mess, suggesting that connectomic similarity is actually a fairly bad predictor of functional similarity, which is kind of a bummer, actually. But this is a harder test for the connectome, so let's give it a break. That said, we have already seen that strong inputs play an important predictive role, so perhaps there is a subset of connections that are going to do better. I used information theory to define an optimal range of connection weights to compute connectivity distance with. And when I do this, we can see that connectivity distance does start to predict functional distance for a subset of the connectomic clusters, which I plotted in the top left quadrant here. However, even in this optimized case, the overall picture is still not particularly impressive, and these results argue that for at least some kinds of functional inference, the connectome alone may never be sufficient. It is also worth noting here, though, that these information-optimized connections turn out once again to be strong inputs, further emphasizing their outsized role in shaping postsynaptic activity. So to conclude on a somewhat speculative note, I think we've learned something pretty cool about the way the brain computes. If you had asked me how postsynaptic integration works before I did this project, I would have said this. Two or more activity patterns, say from cells A and B, are combined to yield some new selectivity in downstream neuron C. We absolutely know that this kind of computation is performed by some cell types in the fly visual system, such as the elementary motion detecting T4 and T5 neurons. However, my results argue that a different kind of computation is pervasive in the brain, where A and B combine only to subtly modify a dominant input, shown here to be neuron A. What's really interesting here is that this style of computation is reminiscent of skip connections built into deep neural networks. Skip connections pass copies of the signals in shallower network layers down to deeper ones. Such connections are required to maintain network stability during training, and all of this raises the intriguing possibility that real brains may face these same stability challenges during development, and that strong connection dominance is the brain's way of solving this problem. I think this is a super cool idea, and I'd love to chat with folks more about how we could test this hypothesis. So thank you all. I'd like to thank my advisor, Tom, and everyone in the lab, but especially those folks who trained me and helped with code along the way. I'd also like to thank Michael Reiser and Sebastian Sung for early access to some of the connectome data. And thank you also to my funders. So I know it's the last day of the meeting. If you can't find me in person to ask questions, please send me an email or check out our preprint. And thank you so much.
Questions?
Hi, yeah. I think it's nice and it's very convenient to be able to principally expect postsynaptic neurons to follow the dominant input. But I'm wondering if you think that maybe behavioral context matters a lot. For example, in the central complex, the primary output classes that people study, these PFL neurons, make up a sort of pitiful fraction of the inputs to the steering descending neurons in the LAL. And it seems like those inputs are much more important, perhaps exclusively in the context of goal-directed navigation. And so do you have any indication in your data that maybe the preference for responding to different subsets of the inputs could be dependent on what the fly is doing?
So yeah, I think that's a really interesting idea. I regretfully did not have a camera on my fly to see exactly what it was doing during my recordings. But we do know of some cases of modulation of activity in a context-dependent way in the visual system. So for example, octopamine boosting the gain of signals. So I absolutely think that that is a piece that is being missed here. And I wish we had done the video recordings to maybe be able to get at that a little bit, but unfortunately it's not part of our data set, yeah.
Thank you.
Thanks.
Great talk. Do you know what the mechanism is? Do you know what's causing one input to typically dominate?
It's a great question. I do not know. I mean, something presumably developmentally is happening that is causing one particular connection or maybe a few to get emphasized and others to get depressed. There's presumably some kind of Hebbian plasticity mechanism, could be one. There is some work that has shown pattern spontaneous activity in the fly visual system as it's developing in the pupa. And I have some preliminary experiments that suggest when you knock down genes responsible for that you get changes in synaptic weights. And I'm hoping to look to see if that actually is changing the connection probabilities, the connection strengths as well. Yeah.
Great talk. This is really cool. So I'm really curious about this connection between the connectome and or the lack of connection between connectome and physiology. And I feel like this is something that we see in C. elegans too, where there's this disconnect. And there's two possible ways this could happen, and I'm curious just your thought on this. One is maybe you could, I don't know what your information preserving metric is, but maybe you could take the data that you already are really confident in and learn some rule based for what you see in the connectome, and then try to predict other neurons. And maybe there is this connection, but we just can't see it because it's not quite as trivial as we hope. The other is in C. elegans, you often see peptides released that vastly modulate the interactions, and that's much slower, but maybe there's something going on similar here. Do you have any thoughts on this? And this is extra connectomic, right?
Yeah, no, I mean, I think similar to the first question, the modulation is a huge piece of what could be going on behind the scenes, and is definitely one of the things that you can absolutely not see in a connectome, pretty much ever. Yeah, and so I think one interest, just in the space of interesting other tests that we could do, If we kind of start with the data and ask, like, can we train a model to see what like connectivity pattern is gonna like actually provide the pattern of activity that we see across you know, the medulla, the optic lobe in general, and then look at the places where that connectivity from that sort of optimized case are failing to match the actual connectivity in the connectome. I think we can start to build a few more insights about why this is failing the way it is. Yeah.
Cool, thanks a lot.
Thanks.
Last quick question.
A relatively simple explanation for this could be that the integration time constant of your neurons is faster than your imaging time constant, such that you have things which are not perfectly synchronous, which you measure. Have you tried such a simple model and exclude it?
So I have not formally tried that model. I will say the imaging rate that I'm imaging at is known. I mean, it's certainly faster than the for neurons in the medulla that have been previously recorded, were definitely faster than that. And so I would suspect that that's probably not the case, but I agree that we should formally build that out, yeah.
Thanks.
Thanks again.
Thanks, Asha. Thanks. Smart thinking.
Just wanted to say hi and introduce myself briefly. I'm Juliana Georgieva. I actually collaborate with